Okay, guys, lecture number 33 and 34 are about the lipid biosynthesis. So I've divided them into parts. The learning objectives for this 133 are we will look at the fatty acid biosynthesis, explain its stoichiometry, describe the fatty acid synthesis for the polyunsaturated fatty acids, DHEs, and the phospholipids. So first up are some major differences between the catabolism of fatty acids, that is breakdown of fatty acids, and anabolism of fatty acids, that is the synthesis of fatty acid, which we are going to discuss in today's lecture. So catabolism or breakdown of fatty acids that we discussed last time produces acetyl-CoA. So acetyl-CoA is its main product. It also produces reducing power in the form of NADH and FADH2 molecules, which are going to give us energy, and it takes place in mitochondria. Whereas the anabolism, which is also called as the synthesis of fatty acids, this time it requires acetyl-CoA. Means acetyl-CoA would be used in the synthesis. Also, it requires another molecule, malonyl-CoA. It also requires reducing power, but it comes from NADPH molecule, which will be providing the electrons, and it takes place in the cytosol in the animals, so that is also known as cytoplasm. Next up is subcellular localization of the lipid metabolism. In simple words, inside the cell where the different pathways of the lipid metabolism are happening. So in the mitochondria, we saw in the previous slide as well, fatty acid oxidation or the breakdown happens. Acetyl-CoA production also happens in the mitochondria. Another thing related with the lipids, the ketone bodies that are also synthesized in the mitochondria. Endoplasmic reticulum, which also contains the ribosomes on them, are mainly responsible for the phospholipid and the steroid or the sterol synthesis. Structures like cholesterol are synthesized in them, plus some fatty acid elongation and alteration also happens in there. Cytoplasm, with regard to the lipid metabolism, is mainly responsible for NADPH production in the pentose phosphate pathway. And very importantly, the fatty acid synthesis that we have to discuss in this lecture also happens in the cytoplasm. And we read in the last lecture as well that some part of the fatty acid oxidation also happens in these organelles known as peroxisomes. So this slide is about the sources of NADPH molecule. As this NADPH molecule is absolutely essential for the synthesis of fatty acid. We cannot synthesize a fatty acid without this NADPH. Cytoplasm or cytosol is a part of the cell where there are high levels of NADPH, and that is one of the main reasons why this fatty acid synthesis also happens in the cytoplasm, because there is enough NADPH here. Now there are two main sources of NADPH in the body. One we already discussed, that is the pentose phosphate pathway. There was a full lecture about it in the carbohydrate metabolism, and NADPH is one of the products of that pathway. And this was the pathway, what it looked like. Glucose was converted into ribulose after 4 or 5 reactions, and we made these NADPH molecules. The second source is this enzyme, malic enzyme, which converts malate into pyruvate and also produce a NADPH molecule. But an important thing to remember is that PPP or the pentose phosphate pathway is the major source, whereas malic enzyme only produces a little bit of NADPH. So in adipocytes, both of these sources provide NADPH, PPP as well as malic enzyme, whereas in hepatocytes and in mammary glands only PPP works, so that is the only source of NADPH in these organs. So to start off this fatty acid synthesis, we need the acetyl-CoA molecules. That is the starting material for the fatty acid synthesis. This acetyl-CoA will be converted to citrate, and citrate goes out for the synthesis of fatty acid. And the process is known as the shuttling of citrate, basically outside of the mitochondria into cytoplasm. And this part as well we discussed when we were discussing in the citric acid cycle. This is known as the amphibolic nature of the citric acid cycle. So as I said in the previous slide, that we need the acetyl-CoA molecule to start the citric acid cycle, and most of this acetyl-CoA is produced in the mitochondria. So it's present in the mitochondria, but fatty acid synthesis must happen in the cytoplasm. Therefore, we will have to transport this acetyl-CoA from the mitochondria into cytoplasm. Acetyl-CoA itself does not have a transporter, so it cannot cross this mitochondrial membrane, and that is why it will first be converted into citrate, as we saw in the previous slide. This is the first reaction of the citric acid cycle, and the citrate can easily go through a transporter, which is known as citrate transporter. So citrate transporter transports this citrate, and the first reaction in which we synthesize citrate is catalyzed by citrate synthase enzyme. So the citrate comes out into the cytoplasm through the citrate transporter, and then it is going to break down to give us the acetyl-CoA molecule again. And this is done by the enzyme citrate lyase, so which will break citrate into acetyl-CoA and the oxaloacetate. So they will be regenerated. This enzyme requires energy, which will be provided by ATP, and now we can use the acetyl-CoA for the lipid biosynthesis. So this is a method that was used earlier for transporting some other compounds as well. We have we have a molecule, say for example A, on one side of the membrane, but it does not have any transporter to move to the other side. So we join it with B and make a complex of AB. AB can be transported through the transporter, and when it goes to the other side, it is broken down again to the A and B, and we can use the A molecule, which in this case is the acetyl-CoA. We don't need the B, which in this case is oxaloacetate, so that will be transported back to the mitochondria. So this is how the whole process looks like diagrammatically. This is the shuttle for the transfer of the acetyl-CoA molecules from mitochondria into the cytoplasm, which we just discussed. So here we have the acetyl-CoA in the mitochondria. Multiple sources are there, and we have to take it to the cytoplasm on the right side. So we joined it with oxaloacetate to make a citrate molecule. Citrate passes through as it has a transporter, the citrate transporter right here in the center, and inside the cytoplasm, citrate will be broken down to acetyl-CoA and oxaloacetate again with the help of lyase enzyme. So synthase is the enzyme that makes citrate, lyase breaks citrate. We have acetyl-CoA, which we can use for the fatty acid synthesis now, and 1 ATP is used by this citrate lyase enzyme. But the oxaloacetate must be taken out back into the mitochondria. So it's converted into malate by the malate dehydrogenase enzyme. Then it has two ways by which it can be transported back into the mitochondria. One way is malate directly goes through the malate alpha ketoglutarate transporter. This is the one, the top one at the bottom, which takes malate out, and malate gets converted back to acetyl-CoA. The other one is known as pyruvate transporter. In this scenario, malate is first converted into pyruvate. Pyruvate goes out through pyruvate transporter, into mitochondria and then converted back to oxaloacetate. And this path uses one more ATP here by the pyruvate carboxylase enzyme. So the main thing to remember is for transferring one acetyl-CoA molecule from the mitochondria to matrix, we will be using two ATP molecules. So the cost is two ATPs, and it will also generate one NADPH molecule as well. So NADPH is going to be produced. So the oxaloacetate, which is present in the cytoplasm and has to be brought back into the mitochondria, as we saw in the diagram, I can go back by two ways, but in both ways it first will be converted to malate. In the first scenario, the malate will be converted into pyruvate and be taken out into mitochondria with the help of the pyruvate transporter, as we saw in the previous slide. Then the pyruvate is going to be converted back to oxaloacetate, and this process requires ATP. The second method was malate is converted to the is directly taken out by this transporter, malate alpha ketoglutarate transporter, and once in the mitochondria it will be converted back to oxaloacetate. So these are the two shuttle systems or two transporters that take oxaloacetate from cytoplasm back into the mitochondria. So once we have the acetyl CoA in the in the cytoplasm, sorry, we can start the process of making these fatty acids. And the first reaction is known as carboxylation of acetyl CoA. Very important reaction in the body, catalyzed by the acetyl CoA carboxylase enzyme, shortened as ACC, which converts acetyl CoA into malonyl CoA. It's a big enzyme. ACC has got three subunits. One is known as biotin carboxylase. The other one is, or the second one is biotin carrying protein, and the third one is trans carboxylase. So these are the three parts of the same enzyme, ACC. The biotin carboxylase, or the first one, mainly attaches the carbon dioxide to the biotin ring, and the second one carries the attached or transports, you can say, the attached carbon dioxide from the biotin carboxylase to the trans carboxylase active site, and the third one, trans carboxylase, transfers carbon dioxide from biotin to acetyl CoA and thus makes the final product malonyl CoA. So this is the acetyl CoA carboxylase, the reaction represented diagrammatically. So simply the reaction is the acetyl CoA converted to malonyl CoA. We add up a carbon dioxide to it basically, and this acetyl CoA is a two carbon compound. So we attached a carbon dioxide to it, and malonyl CoA becomes three carbon compound. So that is one thing to remember. ACC, or acetyl CoA carboxylase, is the enzyme. As we saw, it has three parts. The the biotin carrier protein, the trans carboxylase, and the third one, which takes the carbon dioxide out of it. So the whole process basically is stepwise mentioned or represented on the right side of the diagram as well. So once we have all the required material, we can actually start the process of the fatty acid synthesis, and the main process is done by a single enzyme, FAS1, or the full name fatty acid synthase. Again, it's a big multifunctional enzyme that is mainly going to do the whole process, and it basically catalyzes a repeating four-step sequence. If you remember the beta oxidation, we also had four reactions that were repeated over and over again in that beta oxidation. In four reactions, we took two carbons out of the fatty acid and shortened the chain. So the opposite will happen in fatty acid synthesis. In these four reaction cycle, we will add up two carbons to the going chain until we get to the standard or typical fatty acid, which is the palmitic acid, which has 16 carbons in it. So the overall goal of this enzyme is to attach a 2-carbon acetate unit from the malonyl CoA to the growing chain and then reduce it, basically increase the carbons by 2. And the reaction involves 4 parts of the fatty acid synthase enzyme, or you can say 4 enzyme catalyzed steps are there. First step is condensation, which is attaching 2 molecules together. Second is reduction, taking or loss of electrons. Then dehydration, take a water molecule out. And the fourth one, again reduction, which is again loss of electrons. So this is the 4 reactions or 4 steps that you absolutely need to remember their sequence as well. Just like you needed to remember the beta oxidation steps, the fatty acid synthesis steps are also very important to remember. So first up is condensation, then reduction, dehydration, and in the end one more reduction. Okay guys, next slide is about an important protein known as a acyl carrier protein, and this protein plays an important part in the fatty acid synthesis. The main role of this protein is that it acts as a shuttle or the transporter in the fatty acid synthesis. It delivers the acetate in the first step or in all the remaining steps, malonate to the main enzyme fatty acid synthase. So in other words, it shuttles the growing chain from one active site to the other active site. And this ACP protein, which we can see in the diagram with this brown molecule, is covalently attached to a prosthetic group which is known as phosphopentethine, and which is derived from pantothenic acid, which is the vitamin B5. So from vitamin B5 we derive this group phosphopentethine, which is attached to the ACP protein. And this question has a chance of popping up in the exam as well. Which molecule is attached with the ACP protein? It is the phosphopentethine, which is derived from vitamin B5. So there are 2 thiol groups which contain the sulfur atoms, and we attach these thiol groups with the correct acyl groups before the condensation reaction, that is joining reaction, which is the first one of the 4 reactions that occur in one cycle. So the thiol, one of the thiols is present on the ACP, that is the acyl carrier protein, and another one is present on the cysteine residue of the fatty acid synthase. So in the first reaction, the acetyl group will be transferred to the ACP protein, so it will attach with the ACP proteins, and the malonyl group will be attached with the fatty acid synthase basically. So we can understand the whole process with the help of diagram a little bit better. So here is the ACP or acyl carrier protein, and here is and the other thiol group, which is a part of the fatty acid synthase. This sulfur group, you can say this is the part of fatty acid synthase, and this brown one is the acyl carrier protein, which also has the sulfur or the thiol group. So I've represented them with 1 and 2. So the important thing to remember is the malonyl group or the malonyl CoA attaches with the ACP or acyl carrier protein, which I have designated as 2 here. So malonyl group attaches with ACP, and the acetyl group will be attached with the other sulfur or thiol group. So once we have these 2 groups, so malonyl and acetyl CoA, attached to their respective thiols, then the first reaction, condensation, will take place, and we join these 2 groups together basically, which is the decarboxylation reaction. So we take 2 carbons of this malonyl CoA and attach with the acetyl CoA. So this whole group will be attached with the ACP protein basically at the end of first reaction. So we take the acetyl CoA, and join it with the malonyl CoA in the condensation reaction. Do remember acetyl CoA has 2 carbons in it, whereas malonyl CoA has got 3 carbons. So once we join these two groups together, the result should be a 5 carbon molecule, but instead we get a 4 carbon molecule, and one of the carbons is going to get out or is removed in the form of this carbon dioxide. So in condensation, we join malonyl and acetyl-CoA groups. The resultant product will be attached with the ACP protein, and 1 carbon dioxide goes out. Next step is simply reduction, which is loss of electrons. So NADPH molecule will be the one that will lose the electrons, and electrons are going to be incorporated in this 4 carbon molecule. So step number 3 is dehydration. In other words, removing of a water molecule, which we can see getting removed from the group. So we simply remove the water molecule from this 4 carbon alkyl group that is attached with ACP. And last one is again reduction, similar to the reaction number 2. NADPH will provide the electrons which will be incorporated in this 4 carbon molecule, and basically we have lengthened it by 2 carbons. So we started off with the 2 carbons, and at the end we have got the 4 carbon molecule attached with this ACP protein. And this whole process, all the 4 reactions are done by fatty acid synthase or FAS1 enzyme. So this was the first 4 reactions, or the first cycle that we just saw, joining the 3 carbon malonyl CoA along with the 2 carbon acetyl CoA. At the end of 4 reactions, we got this 4 carbon molecule that is attached here with the thiol of the fatty acid group. So here is the 4 carbon that we got at the end of the 4 reactions. Now this whole process, or the 4 reactions, will be repeated, and several of these cycles will take place. In the next cycle, again we bring a malonyl CoA molecule, which is a 3 carbon molecule, attach it with this ACP protein. These two condense or join together, and this 4 carbon chain at the end of next cycle of 4 reactions will result in this 6 carbon chain that we see in the second step. So in each round of 4 reactions, basically we are adding up 2 carbons at a time. So we started off with the 2 carbon acetyl CoA. After 4 reactions, we got to the 4 carbons, and then 6, and then 8, and then 10, and the process will continue until we get this 16 carbon typical or standard fatty acid, which is known as palmitate, also known as palmitic acid, which has got 16 carbons in it. So this one is considered to be a standard fatty acid. Once we have this standard fatty acid, this will break away from the fatty acid synthase enzyme, and it will be free. If we want to use it, we can use it. Otherwise, we can make any changes to it. We can add up carbons to it, make it a 18 carbon fatty acid, 20, 24, and so on. If you want to add up some double bonds in it, make it a unsaturated fatty acid, we can do all sorts of changes to it. But do remember, in this process of fatty acid synthesis, always first we make this palmitate or palmitic acid, then only we can make some changes to it. And a total of 7 cycles will take place. So this is one round of the 4 reactions, or one cycle. This is the second. This is the third one, which will result in 8 carbon molecule, and then 4 more cycles. So a total of 7 cycles will take place. This word is important to remember. 7 rounds or 7 cycles will produce the palmitate or palmitic acid at the end of this whole process.